Science Officer of our company in North County in Vista, Old Mushroom Superfoods and New Ingredients. She came in as our Chief Science Officer. She's a Canadian person, and so she's got a little bit funny way of talking, but overlook that, please. But she's a real dynamo. She's really turned the company around. We're just, if you get a chance, come get some of her samples out in the booth. They're handing out samples of the products we make. But she's doing some incredible research and really pushing the company in some new directions. And we're in the process of really turning on a new 160,000 square foot facility in Vista. And the mushroom business is finally shrooming. It was a hard sell for years and years and years, but as evidenced by you people coming to learn about mushrooms, people are interested in mushrooms. And this is, she's going to dig into kind of the confusing world of functional mushrooms, which is the best way to take these mushrooms as a therapeutic agent, the different ways of growing them, of processing them and taking them, and pay attention. It's a pretty dense subject, and she's taking on a magical mystery tour of mushrooms. You're going to learn a lot. And
then we have Sam too.
And Sam as well. He's former president of the Mycological Club. He does all of our spawn work and breed and hybridization and strain development, and they make quite a team. In fact, they're enabling me to kind of retire, which is wonderful. I can turn the reins over, bringing in some brains, so I don't have to. I can start to take it easy.
All right, thank you, Steve. Thanks for the kind intro, the kind words, as always. Steve is just such a wonderful founder. You know, sometimes you hear about new companies and quote unquote startups and how intense the founders are, and you know, he's a man full of empathy and bring a lot of peace and joy to our work. And we made these fun T-shirts as well for the company. Anyways, so thank you, Steve. Today I'll present with Sam. And like Steve said, we work together. Sam's our mycology director, and I do other things, science, and dabble in mycology with Sam. And really what we want to talk about today is a little bit of a state of the industry. You know, what mushrooms are we using for functional ingredients? How are we picking strains? How are we testing the mushrooms to know whether or not they're efficacious? And it might be surprising that there's still a lot to discover. And so today it's a little bit about the challenging discoveries that we make and that bring more questions than answers sometimes. So we'll try to share a little bit of that and how we do science. We're also going to talk about some of our growing trials and how it's relevant for the efficacy of the product. So our company is Ohm and M2 Ingredients. So we sell bulk ingredients to other brands, and then we have our own brand. And the company was founded by Sandra and Steve. And on the right is a picture of our new facility, which is huge. It's like 3 Costco's full of mushrooms. All right. Well, this was working in the test, but oh, there it is. So we are vertically integrated, meaning we create our own strains, we produce our own spawn, grow our own mushrooms, dry them, and make finished products. So everything is done in our facility. And that allows us to really control the conditions the mushrooms are grown in, but also control our substrate and the type of strains that we use. These are just some of the species we grow. We grow 10 different species. And so we have our own strains of all of these. And so why this interest in mushrooms, and how is it relevant to finding functional ingredients? So my background is in natural product chemistry, and we looked in nature for things that couldn't defend themselves physically because generally they had very interesting chemistry and could be drug leads or new functional ingredients. So things that don't have a shell or spines or that aren't hard. So mushrooms really fit the bill for that, right? And when you think about mycelium, mycelium is one cell layer deep, right? Wide. So if you could think of your skin only having one cell layer, anything could essentially penetrate in, cause an infection. So the mycelium is essentially waging a chemical warfare on its environment, right? Also digesting its environment. But because of that, it creates really, really interesting chemistry. And so fungi are an amazing source of new chemistry because first, there's a lot of different species, right? High biodiversity compared to plant. Potentially 1.5 million species, we're not sure yet. That keeps changing. Oops, and whereas plants only have 250,000 species in that kingdom. So sheer diversity means diversity of chemistry, but also because these organisms can't defend themselves physically, they tend to have really interesting chemistry. So lots of interest in drug discovery with fungi, but also in functional ingredients, and that's mostly guided through traditional medicine. So there's a kind of a lot of talk on social media right now around mycelium versus fruit body and what's better and what's worse. And really, it all depends. It depends on the strain you're talking about, the species you're talking about, how it was grown. It's a little bit like saying, with plants, you should only use seeds or you should only use roots. Everything else is garbage. Obviously, that's not true. It depends on the species, on the plant, and so on and so forth. So there's definitely benefit to both stage of the life cycle. And you'll see a lot of products on the market will have fruit body only. Some will be mycelium only, cultivated in liquid culture. Some will be solid fermentation culture. And then you'll have products with both fruit body and mycelium in solid state culture, and that's what we grow. So on the bottom right here is our Cordyceps. So this is one of our really exciting strain that Sam developed. It's a real producer, and it's a beautiful, beautiful mushroom. All right, so we talked about mycelium and fruit body and why, what should we use, what's relevant. So if we just take a look at lion's mane, and lion's mane has a number of different molecules that are important for its biological activity. There's 4 main classes, so the beta-glucans, which we find in all mushrooms, so immunomodulating molecules. But you also have the ericinones, the erinacines, and the ericerins, which is a new class of molecule discovered in 2023 in the most popular and most probably widely grown functional mushroom today in the industry. So we're still discovering new things in really, really common mushrooms. And so what we see is that some of these molecules are only in the fruit body or only in the mycelium. So if you only have one of those parts for lion's mane, you might be missing really key chemistry for its biological activity. The other thing to consider is when you're buying a product and you have a water extract or an alcohol extract, only the molecules, the chemistry that's soluble in that will be found in that product. Whereas we sell a full spectrum powder where you'll have the full chemistry that the mushroom will produce, maybe in lower levels than if you concentrate in an extract, but we believe that there's so much interesting chemistry in these organisms that we sell the product as a full spectrum powder. Did I skip a slide? No. All right, so right now, when we make a decision, so Sam and I and the team will be sitting there and like, well, this Cordyceps strain looks amazing. It's got huge fruit bodies, it's dark orange, must be good. But is it really? Do we know if it's more efficacious than the straggly looking paler one? Perhaps that one is better, right? And is it the mycelium of this strain versus the fruit body of this strain that's better? So we need some tools to make a decision. And ultimately what we want is an efficacious product. And the best way to see is it efficacious or not is through a clinical trial. Unfortunately, despite Steve's great business success, we don't have an unlimited budget for clinical trials. So we need other ways to answer those questions. And so we need to look for different chemistry in different parts of the mushrooms or in the different strains to compare them and understand how efficacious they could be. The problem with that is that there's few studies that actually link specific molecule to a benefit. Often we'll say, for reishi, for example, ganoderic acid A. There's about 20 ganoderic acid. A was the first one to be discovered. It's often the one that we talk about because it was the first one to be discovered, and there's a test for it. And so whenever there's a clinical trial that's done, they'll say this product has X amount of ganoderic acid A. We don't really know if that's what's really causing the benefit of reishi, but that's what's mostly talked about. So there's a real need for more science and understanding what these molecules do and are they actually related to the efficacy of the product. So today, from a lab, so if we call up a lab that specializes in mushrooms, only the molecules in green there would be offered as a standard test. Nothing else is actually offered commercially and in a repeatable manner. So that makes it super challenging for us to make any kind of decision. So we looked for new methods or not necessarily new, but pretty new methods to find what chemistry is in the product and how can we make decisions. So we decided to delve into metabolomics. So essentially, we partner with a lab that has a very sensitive instrument. It's called a time-of-flight mass spec, and we could look at all the chemicals that are present in one sample. So essentially, that method will scan the sample for molecular mass at a very high level of resolution. And then it'll match it to a database that has known molecules, so they're natural product databases, and it'll match it to that with a high level of certainty. So after that, we can say, okay, that sample contains aranacine A, B, C, D, E, and other things. And so we use that method to actually compare different strains together to understand, like, is the chemistry more interesting? Is there more of certain molecules? It's a very expensive method, but to drive decision on what could be more efficacious, it's a really good first step. And then we follow that with cell-based assays and clinicals. So let's talk about the case study of lion's mane, so Ericium. So we'll talk about the strain selection that we did. So what is the best strain that we could grow? We'll talk about the fungal parts and the work we did in metabolomics there. The work that we did in metabolomics to pick our drying temperature, right? Because lion's mane has really, really sensitive chemistry, so sensitive to heat, and we don't want to dry it too hot and degrade it. But we also want to cook the mushroom and open its cell wall, right? Break up that chitinous cell wall. And then we'll talk about the types of products and the comparison there. Sam, do you want to cover some of the work you did here?
So in order to do these studies and to come up with a variety of samples and different strains that we can test and figure out, which ones are best to use, we have to collect a bunch of different strains. We have to collect a bunch of different strains and then evaluate all these strains, and it takes a long time. It took us a whole year to do Cordyceps and lion's mane. When we start, we'll either start from a spore and we'll isolate a bunch of different cultures and cross them together. Sometimes we just collect a bunch of different strains where in the case of lion's mane, there is a numerous amount of strains on the market and a lot of people have different ones. What we do is we grow them in their natural, like a more controlled, more natural environment, like they would in nature on a bag, and we determine the different phenotypes that are available and whether or not these different phenotypes exhibit different traits that are going to be good for growing in the methods that we use. And for instance, in the lion's mane, we found that European strains tended to have a more spiny fruit body development, whereas some of the East Coast United States strains had these very compact fruit bodies that didn't oxidize very well. I don't know if any of you have found any lion's mane in nature. You'll usually see like a lot of browning occurring on the top of the fruit body. Well, this is due to polyphenols oxidizing on top of the fruiting body. And when the spines are exposed, they're exposed more. I'm sure all of you have seen in the news just recently, they're talking about bananas and putting all your fruit in there and making smoothies. Well, you were losing a lot of the antioxidant potential. Well, same thing happening here. So what we do is we grow these strains out. We find which desirable traits that we want that are going to give us the best product. And then we test them in the methods that we use to produce them commercially. And what we found was that these compact fruiting bodies tended to produce very large masses of fruit body development on top of the bags. And this is a nice trait because the ones that don't do this tend to fruit a bunch of numerous fruit bodies around the edges of the bags, which is not what we want. We want a large mass, a lot of fruit body potential, along with the mycelium below. Go ahead. So we also have to go through and we have to determine what light is required, what temperatures, how much food we need to give it, and things like CO2, humidity, all that stuff plays a huge role in the final product, especially environmental controls. Environmental controls, if they swing out too much, they can cause the final product not to produce a certain amount of compounds that we're looking for.
All right. Thanks, Sam. So in the case of lion's mane, we talked about four groups of molecules, right? Beta-glucans, but also the ericinone and erinacines. So ericinones and erinacines were described several decades ago, and there's been a lot of research on them, and they really support the peripheral nerve health. Whereas the ericerin, this new group of molecule, is very targeted to the central nervous system. So having all these classes of molecule in the product will ensure that you get the full benefit that lion's mane can give you. And what's interesting is that these molecules aren't found in the same fungal parts, and they're also not very soluble in water. So choosing the right solvent when you're making an extract is really important. So this is some of the work that we did with our metabolomics tool. So we looked for all the molecules present in the mushroom, but we have focused in on these three classes of molecules. And what we saw is, as expected, lots of erinacines in the mycelium, and then ericerins and ericinone mostly in the fruit body. So that confirmed, what we expected, and that was true for our strains as well. Then we compared a whole mushroom product, so not extracted, so on the right. And these are average height ratio, meaning the quantity of each of those comparison. And then the fruit body water extract had very little ericinone, isoericerins, and erinacines. And that's because they're not very soluble in water. That water extract had a lot of beta-glucans, obviously because beta-glucans are soluble in water. So picking the right solvent when you want to make an efficacious product is super important. We also looked at other molecules. So we were very surprised to find some chemistry that hadn't been described in lion's mane during this exercise. So we actually found isoramnitine, which is a molecule that's been first described in ginkgo biloba and also recognized for its cognitive benefits. So that was really surprising and exciting to see it in lion's mane. And right now we're trying to figure out, is it because we're growing it on oats? Is it something in oats that the mycelium is converting and making this new chemistry? So very exciting. And what we found was that, again, we saw a lot more isoramnitine or phenylenol and armanol, other plant antioxidants that we didn't expect in lion's mane are much more concentrated in the unextracted whole mushroom powder than in the extract. And again, it's because water isn't a great solvent for these molecules. We also use this method to decide which drying temperature we should use for lion's mane. So 160 wasn't hot enough to essentially break up the cell, make this chemistry available. But at 190, we got lots of those chemicals released, and so providing a nice efficacious product. Okay, so what does it all mean? So this was lion's mane, and this was a few of our strains, a few of our processing methods. So you can see how confusing this all is and how many questions there is to answer. So the kingdom of fungi is very vast, and there's a lot of species, and within those species there's a lot of strains and a lot of differences there. So very important to do your own work and when you're making these preparations to really drive to efficacy. In lion's mane, both fungal parts, mycelium and fruit body, are key for the biological activity to get the full potential that lion's mane can offer. And also picking the right solvent or the right preparation method will be key for efficacy. So we'll move into Cordyceps, where we made a similar exercise, and we'll talk about the growing and the chemistry as well.
Yes.
This is our grow room full of Cordyceps.
Nice.
So Cordyceps is unique fungi. It's a short-lived fungus, meaning that it is, once it starts from spore, it basically fruits and it's done. So it's a really difficult fungus to work with and maintain strains on. So we have to find unique methods to keep them vigorous and preserve those qualities that we want. It was a really interesting project. We ran Cordyceps on a huge amount of different types of media, different amino acids, nucleosides, all kinds of things, just see how it grew. And then we had to determine what the best method was to grow it and produce the most amount of compounds like cordycepin, which is the most desirable other than beta-glucans in the Cordyceps. There's others I'm sure Julie might talk about, but this is the one we had focus on. So when we grew it, we've discovered that when we give it more food and we select our strains on that quantity of food, we are able to produce large amounts of cordycepin per square footage of our space. You can see in like the graph here on the substrate, per cubic foot, we were actually able to produce 5,340 milligrams of cordycepin as opposed to 476 in tray culture. And this is due to the amount of food that's available to it. Now, you'll say, well, you'll see most people growing in trays on a thin substrate. Well, that limits the amount of cordycepin we can produce because there is the cordycepin is actually produced and started inside of the mycelium. So if there's not a huge volume of mycelium to start off with, you end up with a bunch of fruit bodies, but you end up with less cordycepin in it. So the fruiting bodies will have less cordycepin per fruit body than it would if you had more substrate in it. So what we did is we evaluated some strains we had. We got the best perimeters that we could choose. We determined the type of light mixture that would produce the most amount of compounds in the mushroom. And turns out that we found that higher CO2 levels at the beginning actually produced more cordycepin at the end.
By accident.
By accident, yeah. So we had this room. We started Trametes and Cordyceps in it at the same time. And so Trametes versicolor, turkey tail, produces huge amounts of CO2 when it's growing, lots. So much that we have to open up the air dampers and get it out of the room because people will just pass out in the room if they walk in there, right? And so when we paired these two up together, we did some testing on the fruiting bodies, and we actually ended up with 17.2 milligrams per gram in the fruiting bodies, which was enormous, comparable to other commercial fruiting bodies, which usually are around 3, maybe at a max around 7 if you have a really good grower. So we ended up discovering that that was a good way to produce a lot of cordycepin at the end, was by like getting the CO2 levels up. So in order to do this, we had to collect a bunch of different strains, isolate the spores, and grow each individual spore into a monokaryotic culture, which is basically just has one nuclei in each cell. It needs to pair up with another compatible hyphae to produce the fruiting bodies, right? So we started with a grid. We produced 132 different crosses and evaluated all these crosses in the perimeters that we came up with at the very beginning. And we came out with some really impressive strains. We had an outlier that was very consistent at producing 10.5 milligrams per gram, which was really great. And so what we did is we crossed all of these, we grew them out in different size bags, and then we tested them for different compounds and determined which one was going to be the best fruiting strain for us, and you can see them here. We also fruit them in the tray culture to make decisions, so we can compare the results back to the standard way that most people grow it. Next.
I think it's back to me.
Yeah.
So we had all these crosses, all these new strains. Some of them looked amazing, but we didn't know if their chemistry would be reflective of their phenotype. So then we went ahead and tested these strains in the metabolomics assay, and we saw that M2116 was a champion in making some of the cordycepins and cordycepin analogs. So that was really interesting to compare, and that was also the strain that we were most excited about, so that worked out, I guess. That confirmed it. And then we also compared what if we offered an extract of cordyceps versus our whole mushroom powder. And what we found is that in the extract we were getting a lot more of the cordycepin analogs, the pentostatin and the adenosine, but not as much cordycepin. So we're like, okay, that validated our decision to have a whole mushroom product. So now we know the chemistry is there, but does it actually make any difference in people's lives? So again, we don't have the massive budgets yet, so we go into cell-based assays to get a sense from, on a biological level, are we getting an effect from these decisions we're about to make? So we did, our first assay was to look at antioxidant potential of some of our strains of mushrooms. And we did a cell-based assay where the actual product needed to go inside the cell, right? And the cell would be protected from the inside from free radicals. So we wanted to make sure that whatever we were giving the cells would actually cross the cell barrier and be effective in a human model. So we tested our strains against a commercially available blueberry extracts, right? And we think about berries as being antioxidants. We add them in our smoothies, and we look at mushrooms, and they're pale and brown, and how could they be antioxidant? Well, it turns out they're a great source of antioxidants. And we see that here. So our Cordyceps champion strain M2116 was outcompeting blueberry extract in the antioxidant assay. So that was super exciting. And we also saw that our lion's mane was also at high antioxidant potential, which was surprising to me because as a chemist, often the antioxidant molecules are very brightly colored. So cordyceps is bright orange, just like astaxanthin, right? Bright orange, carotene, bright orange, all antioxidant. But lion's mane is just pale, you know. It doesn't seem like it's got any kind of protection from free radical, but it does. So that was exciting results and also confirm what we saw from the chemistry in terms of bioactivity. We also looked at other cellular assays such as metabolic energy. So we looked at capacity of products to promote mitochondrial energy. So why is this relevant? Well, you've probably heard of cold water baths, right? And fasting and all that crazy biohacking stuff. So what you're trying to do there is promote mitochondrial health, promote mitochondrial energy, and some molecules in nature can also do that. And so we tested our Cordyceps strain in lion's mane and saw that, in fact, they did promote mitochondrial energy in the cell, which is a process that slows down as we age, right? And so that was also exciting results. So these are ways that aren't quite yet a clinical trial that we can use to see, is the product working or not? Are we making the right decision in picking the strain or this processing temperature? All right, so this is just a quick glimpse in all the confusing questions we're faced with every day, and we just wanted to, start a conversation and give you guys a sense of the complexity and how much is still to be discovered. I mean, to discover new chemistry in lion's mane in 2023 is kind of crazy. You know, I thought I was born too late, that all the cool discoveries had been done, had already happened, and turns out it's happening now. Yes?
Question on the cordyceps. So I just found out that the actual fruiting body is on top of...
It's actually, so you're seeing the stroma, and the actual fruiting bodies are the little red dots on top.
So was there any distinguishable characteristics then between the strains and how much?
Yes, some of them actually don't actually produce spores. Some of them actually will just make perithecia that are just barren. I mean, it's very variable. Like, and you have to do microscopy work. You have to look at them, make sure that they're actually, you can keep going with it. You can produce spores and keep producing different strains. And that's important because, like I said, it's a short-lived fungus. Some of the strains will only last about a year before, we do cryogenic culture, but that's not a guarantee that later on that it's going to produce well. So what we do is we preserve our isolates and we cross them. We want to get some hybrid vigor into them. So if we see that the fruitings start to fall off, we recross them and go again.
It's very pesky.
Yeah, it's very troublesome fungus to work with.
It was easy, everybody to do it, right?
Yeah.
And I also went to another presentation earlier in the year about the dual extraction process and how not, like you had mentioned, each type of extract is different or the qualities will be different. Have you gone into the different extraction methods then?
Yeah, I mean, dual extraction is usually alcohol and water. So that's interesting because alcohol is a bit better than water at pulling out non-polar chemistry. So not like more fat-soluble chemistry, but it's not that good. It's still very polar. So like in pure natural product chemistry, we'd use like hexane or CO2 extraction to pull that chemistry out. But obviously you can't use hexane for like a product, for a whole product, right? that doesn't work because that leaves all kinds of residues. So it's really hard to pull those fat-soluble like terpenes and things like that out in a water alcohol extract. Yeah.
And then in your products, the mycelium then, is there like a combination ratio that you actually put into your products as far as the whole mushroom is concerned then?
Yeah, so we harvest essentially so we plant the mycelium, it will colonize the grain and then digest the grain and start fruiting, usually as resources becomes more scarce. I mean, it really depends on the species, like lion's mane starts fruiting pretty much right away. But then we'll collect that whole mycelium block and the fruit body on top, and that's the product.
Process everything.
Nice.
Yeah, all the time frames for the different species are different, and we try to adhere to those specific time frames when we harvest.
Yeah. Yes.
I find it fascinating when you're mentioning the lion's mane, some certain compounds are being more soluble with the alcohol extracts versus the water. Being that a lot of the products on the market, being your Ganoderma coffees and lion's mane coffee, you're expecting that you're going to get those compounds out of hot water. Has that shifted your guys' focus in terms of what products that you're putting out or how you want to?
Yeah, I mean, we offer a whole mushroom powder, so it creates a bit of a mud in the coffee. It's not fully soluble, so we recommend finishing the whole cup for that reason. But I mean, traditionally, all these species were often done as a decoction in hot water, and then you'd filter out the filtrate, so you wouldn't necessarily get everything. But in our product, we include essentially the whole mushroom. So what that means is it might be less concentrated than an extract. Like I said, if we would have looked at beta-glucans on some of these, the beta-glucans in the water extract would be a lot higher than in our product because beta-glucans are super soluble in water. But it means that we have essentially all the chemistry the mushroom is producing. We're not leaving anything behind. So although it might be less concentrated, it's more holistic. Yes?
So when you were talking about the Ganoderma and the ganoderic acid A and then the BC, I mean, you work on isolating each acid and I guess seeing what they do, but then do you measure, I mean, you're doing the whole mushroom because it seems to me even if we isolate different compounds, we don't necessarily know how they're interacting with each other.
Exactly.
It's a holistic, you know, so would, you know, A work with just C or, so,
Well,
I mean, I guess it's good to isolate what they are, but continue to grow the whole mushroom because you want that whole interaction.
Yeah, absolutely. I think for us, and we totally agree with that, also the second question is we don't know which one of these is effective for a specific benefit, right? Is A better than C, D, and E? I mean, we spent like weeks and weeks combing the literature trying to figure out like, okay, yes, they tested with a ganoderic acid A extract, but we don't know if that was the only thing in there or how it was processed. And so we don't really know if that was why there was an effect. So the reason we care about that is it's really hard to make decisions just based on efficacy because it would be too expensive. Like we can't do 200 cell baseline assay or 200 clinical trial to make a decision. So having a chemistry tool would be really useful. Unfortunately, the labs are not there, so that's why we turn to metabolomics because we can see essentially like all the chemistry at once. And it won't tell us how much is in there, but it'll tell us, compared to something else, how much more is in there. It's like qualitative, and it allows us to compare different samples together. Yes?
I'm just wondering, have you come across anything that, if I'm taking these powders regularly, is there anything out there that you can add to increase bioavailability?
Yes, but it depends on the species and the molecule. I mean, that's something that we're investigating currently on some of our species. I don't know, Steve, if you want to talk about something specific.
There's been some studies showing taking it with vitamin C increases the uptake, but I wouldn't say those are conclusive studies by any means. It's kind of almost hearsay, kind of you see it on the web and whatnot. People say it, but I don't know what they're basing that on. I don't think anything like pure from the black pepper. I don't think that helps much with the fungal compounds. But that'd be a good research study.
Yes.
So you mentioned that the higher temperature for lion's mane made the extraction easier, or
Yeah.
you were able to see more of the active molecules. Is that similar for other mushrooms like Ganoderma or Cordyceps?
Yeah, so it definitely does vary by species. So 190 was the optimal temperature. At 210, we saw some degradation again. So for that one, yes. Typically, you don't want really high heat. You want enough heat and moisture to essentially break the cell wall open. And that's what we saw. I think we're hypothesizing that at 160, we weren't breaking the cell wall enough. And that's why we weren't seeing some of those molecules in that sample.
And was that the temperature you're extracting at, or you're drying the mushroom?
Drying at, yeah.
Extractions. And that was in contradiction to a published study, a peer-reviewed study, where they showed that at 160 and above, it denatured the RNA.
I mean, it's all about time under temperature and like what type of oven and yeah, again. Yeah.
So you would in an oven then dry with some humidity enforced, or you would cook it and then
At the very beginning.
At the
Yeah.
very beginning, so the oven is closed essentially, so it builds up a lot of moisture, and it essentially like steam.
Okay.
Steam cooks it.
Yeah, steam cooks it. Yeah, and then we open the vent, and then the mushroom dries.
Gotcha.
Yes.
Has anybody combined with lion's mane, the other type of herbs that work for the brain, like the Ayurvedic herbs, or some Ayurvedic herbs that are for the brain? Do they work in combination as well with each other?
I know there's several formulas out there on the market. I mean, ashwagandha is super popular right now, so like we pair it with reishi, but not with lion's mane. I wouldn't be able to speak to that specifically. Steve, do you have any?
Yeah, we're looking at combining it with some other nootropic compounds, but our marketing department tends to go wild on stuff. We don't want to give them too many ideas.
We're trying to be good at mushrooms.
We're trying to be good at mushrooms, exactly. Yeah, because we have to sell, we outsource all the other compounds, the other ingredients. So we like to be more of a mushroom company, but it's you do what you can.
You had a question?
Yeah, so I'm assuming your products are regulated as a supplement. Is that true? So what steps do you take to ensure the safety of your products?
Yeah, so lots. We grow under GMP practices. We're actually a BRC AA rated facility. BRC is a certification of food safety and quality. It's the most rigorous certification. So not only do we do FDA GMP, but we go definitely several steps above and follow the BRC standard. So all of our environment in the facility is controlled. When the substrate comes in, it's oats, organic oats, and we test them for any residue of pesticide, herbicide, aflatoxin, any kind of mycotoxin as well, to make sure that the substrate is really clean. We test for heavy metals as well. And then we essentially autoclave the substrate so that it's fully sterilized before we plant it. So 2 reasons. Food safety reason, but also to grow pure cultures. So all of our cultures are DNA tested for integrity to make sure we have the right species and also ID our strains. And then all the facility is controlled. So no one that doesn't work there can go in and out, and all under control conditions. And then we test the final products as well for micro-contaminant, heavy metal, and we do also some validation on pesticides, sulfites, gluten, all those things. And then in terms of safety, like in terms of eating the product and what the safety is there, we have done some toxicology studies, both in cells and rats. So GRAS is essentially the certification, not a certification, but a status from the government, the FDA, that tells you whether a product is safe as a food or not. And they have very standard, a battery of tests that you have to run that are very standard. So we've done that. We couldn't feed the animal enough mushrooms. We got to the equivalent of 27 grams a day, like what we would have to consume. And they couldn't eat anymore. So we don't have an LD50 yet because we couldn't fit it. Yeah. So those are the type of safety studies we've done. And we've also just completed 2 clinical trials with 400 participants in one and 500 in the other, and we didn't have any different adverse event in the placebo versus control. We've got lots of with some GI issues in both group. Maybe it's the fiber piece, I don't know. Yeah.
With the GI adverse effects, you said that you grow on oats. Does the mycelium break down gluten then in wheat-based grain?
Yeah, so
growing grain?
Oats actually don't contain gluten, but they often are cross-contaminated with gluten because the silos that hold the grain are the same for oats. But we have a dedicated supply chain for gluten-free oats, and then we test our incoming substrate for gluten, and we test the finished product for gluten as well.
Do you know on the other side, though, do wheat berries if mushrooms, edible mushrooms, were grown on the wheat berries or substrate, do the mushrooms actually break down that gluten? So it's not in the final.
Well, they don't. It's not transferred. I know a variety of people who grow on other substrates, and people don't seem to have any issues with eating the mushrooms, but they would if they ate the bottom part.
It breaks down the gluten to a degree, but not all of it. So if you're consuming the mycelium and the myceliated substrate, if it's grown on wheat, there'd still be a fair amount of gluten there.
And I know that there's ancient wheat that doesn't have as much gluten, or the proteins are different pre-World War II wheat than today's wheat.
It'd be better, yeah.
One thing to watch out for if you're producing mushrooms and testing for gluten, that's another pickle. There are terpenes that interact with the gluten test in mushroom, and so you might get false positives, which is very confusing when you're trying to figure out where it's coming from. But it's not gluten. But there is a test out there called R Biofarm, and that's specific to product like oat-containing mushrooms and mushroom products. So where you don't get those false positive. Yeah.
It's bloody complicated.
Yes?
Are you guys utilizing any kind of targeted editing technique like CRISPR?
No.
No.
Thank you.
We're interested. I mean, I'm personally interested in things like that, but no. No.
Are like the targets even like known enough at this point to go after that sort of thing?
Yeah, I mean, I think it's definitely something that could be considered. We're very, because we're organic and non-GMO and all that, we're kind of limited in the type of work we can do. I mean, we can do PCR to look for specific genes and so on when we're doing the mating. It's not a GMO technique. It's more to know what's in the strain. But yeah, we're a bit limited because of our certifications. Yes?
So you use oats as a substrate for all 11 species that you grow?
So, and none of these species really evolved to grow on oats, so it takes us a while to find a strain that's...
Yeah, not all the strains will actually grow very well on the oats, so we have to select for it.
Is it an oat seed?
It's oats with the hole on it. The hole's in.
Hold on.
Yep.
You don't germinate it to get endosperm or anything active before you do it?
No.
See, I thought that oats have gliadin in it. That's the oat protein. And wheat, gluten is wheat protein.
Yes.
So is that, but they kind of affect people.
No, it's adenine, not gliadin. It's a different
Gliadin?
Yeah, it's a different protein.
And that kind of affects people the same way that gluten intolerance, though, or...
Generally not. Most people, unless it's really severe, they're fine with oat.
Okay. Yeah. Another question. You said something about cold baths. How does that?
Yeah. So people, there's a huge trend right now in metabolic health at the cell level, right? And how to, slow down aging and things like that. And people look for things that would activate autophagy, which is cellular pathways that usually helps your cell survive, like will kick into survival mode and starts recycling a lot of the metabolic waste in the cell.
So that's like hot and cold.
Yeah, so the cold water bath is a shock, right? Your body is like, oh my God, like, because it essentially forces your cell to go into quote unquote survival mode. So they'll start eating, like you'll start recycling in your cells any type of metabolic waste that's in there. So that's a process called autophagy. So you're eating your own cells. But it helps because then that material is no longer floating around and getting in between strands of DNA and creating mutation and creating, slowing down essentially the way your cell work. The mitochondrial, yeah, so it'll activate your mitochondria. Yes?
I might have missed the first part. When you're generating your
your spawn, your liquid culture, to knock the spawn. Have you done any work with like just putting it into a fermenter? You know, you have these industrial fermentations to very large size. And what would be the outcome of doing very high density fermentations just in the absence of substrate? And is there any sort of thought about that?
Well, of course, you're not producing the fruiting body, so you're limiting yourself for compounds that would only show up in the fruiting body. So we're not. I mean, we're kind of looking into some of that from a formulation perspective, but no. We do some fermentation in the labs, but that's strictly R&D.
Some companies do.
As a precursor to solid-state fermentation, is that also?
I'm sorry, what was it?
Yeah, so we do liquid spawn on some species instead of grain spawn to plant the material. So that'll be the pre-step. But there are some products on the market that are liquid fermentation products. So CS4 is a Cordyceps sinensis ingredient that you'll find on the market, and that's done in bioreactor tanks. And essentially
you get the pure mycelium that way, but you lose all the extracellular enzymes and secondary metabolites of the mycelium was put into the environment, into the liquid in this case. You can collect the pure mycelium, you don't have any of the remnants of substrate, but you also left a lot behind in the liquids.
Yes?
Maybe you covered this early. The M2 extraction process, is that a proprietary process that you guys created?
No, there is no such process.
There is no such process. Okay.
We don't extract. It's a whole food, full life cycle product.
Okay.
We steam cook them to make it bioavailable and digestible, but we don't extract. We don't try to concentrate.
You don't try and
We don't think we're smart enough to pick the best thing and leave the other things out.
Leave the whole
Everything
thing.
that's nice.
Got it. Okay, thanks.
Wow, lots of good questions. Amazing crowd. All right.
Three months ago, I would not have followed that.